Photosynthesis, the process how the light energy turns into chemical potential energy. The light energy plus carbon dioxide plus water will yield to carbohydrate, glucose plus oxygen and water. Balancing the equation, the carbon that is in this carbon dioxide will show up in the sugar, the oxygen in the sugar and in the water, the hydrogen showing up over there, and the oxygen is going, the oxygen of the water will be the byproduct of photosynthesis. The carbon dioxide is going to go through reduction and turns into sugar. It is an oxidizing agent, and the water is going to get oxidized, sorry for the mistake, gets oxidized and it's mistake, and it is a reducing agent, going to reduce the carbon dioxide to sugar. The whole thing, so light energy to chemical potential energy. The chemical potential energy is in the bonds in the sugar molecule. This is the chloroplast, and these are the thylakoids. Together makes up the grana, and this is where the light-dependent reaction takes place. Those are the chlorophylls, the pigments that absorbs the light. Blue light absorbed, red light absorbed. The water inside of the thylakoid membrane splits into electrons, hydrogens, and oxygen. So the water goes in, and the oxygen is the byproduct, leaves the plant through the stoma. The high energy electron carriers, the NADP plus, gets reduced by the electrons and picks up the proton too, and carries the high energy electrons and protons to the light independent reaction place, where it's called the Calvin cycle, as well as ATP is going to get generated as through chemiosmosis. The oxidized NADPH, the NADP plus, goes back into the light dependent place, as well as the ADP to get reduced again or charged with a phosphate. So the Calvin cycle happens in the stroma, and the carbon dioxide gets into the light independent reaction and yields to a triose phosphate, a 3-carbon molecule, carbohydrate, and this triose phosphate is going to turn into different carbohydrates, disaccharides, for example the sucrose, or starch as a storage material, cellulose for cell wall, or monosaccharides for cellular respiration. So that is, the triose phosphate will turn into glucose, or one of the carbs. The pigments are materials that they absorb light. The light is an electromagnetic radiation. It contains energy. This acts as a wave, and at the right, actually at the left, high frequency, as we go towards the light, to the right, the frequency will decrease. The different segments are the gamma ray, high frequency, after comes the X-ray, UV, visible light, infrared, microwave, and radio waves. The visible light spectrum is the spectrum that the plants use. the red, orange, yellow, green, blue, and violet. If this line is the energy, the radio wave has low energy and high energy. For the gamma ray, wavelength is between the two crests or two troughs, consecutive ones, and the distance between them. And frequency is inversely relates to the wavelength. So light acts as a wave, like we draw it out, and also acts as a particle like photons. The pigments absorb light, and the absorbed light makes the work. Contains magnesium, the chlorophyll, and the pigments can be carotenoids, and they can be chlorophylls. The carotenoids absorb blue-violet light, and based on what color they reflect, they have different colors, and the chlorophylls are absorbing blue, violet, and red lights, or photons. Absorption spectrum. Each pigment contains an absorption spectrum, and we can use the spectrophotometer to measure the absorption. The light comes from a machine, the pigment is in the test tube, and the transmitted light at each wavelength is going to be measured. The machine will give you a graph. That's the independent is the wavelength, and the dependent variable is the percent of the absorption. On the left is the blue light, and towards the right is the red light. And the chlorophyll's absorption spectrum is absorbs the blue and the red light. Blue is around 400 nanometers, and the red is 700 nanometers. So the absorbed light is that makes the work. If the light is not absorbed, it will not make work, and it will give only the color of the object or the chemical. Engelmann had an experiment in 1883, and he checked what kind of a light do the pigments absorb. What he did, put algae in a row, and he just said if photosynthesis happens, oxygen will be released. Aerobic bacteria went on the other side of the algae, and the opposite side he used different lights to illuminate the algae, and he noticed that the aerobic bacteria started to reproduce and thrive where the red light illuminated the algae, as well as where the blue light illuminated the algae. So with that he proved that these are the two lights what the photosynthesis uses. Covering the light-dependent reaction, here is the thylakoid membrane. Inside of the thylakoid and outside, the other one is the stroma, we have the photosystem here. The photosystem contains the light harvesting complex that contains pigments other than chlorophyll A. And inside, this is the photosystem 2 at 680 nanometer wavelength. Inside of it is the reaction center. The reaction center at the bottom contains the chlorophyll A, and at the top is the primary electron acceptor. The inside of the light harvesting complex, there are the accessory pigments, the carotenoids and chlorophyll B, and they absorb the light and they pass the light energy to each other and excite their electrons. For example, the green does not cause any excitement for the electrons, and the energy gathered will excite the electron of the chlorophyll A. Inside of the thylakoid, the water splits into two electrons, two hydrogens, and oxygen molecule. And as the electron of the chlorophyll A gets excited and will be catched by the primary electron acceptor, the water's electrons are going to replace that lost electron of the chlorophyll A. The electron will be passed on the electrotransport chain, and the energy of the electron along the chain is going to be used to build up a proton gradient, that moving protons from the stroma into the inside of the thylakoid. Pay attention that the pH is low inside of the thylakoid. Here comes the second photosystem, and it has a reaction center too. Next to the photosystem 1 is attached the NADP plus reductase, an enzyme. Light again hits the light harvesting complex. The energy will be passed on, and the electron is going to get excited for the chlorophyll. Electron acceptor is going to accept that, and that electron is going to be replaced by the electrotransport chain. The NADP plus reductase is going to reduce the NADP plus to NADPH, and the accumulated hydrogen ions inside of the thylakoid are going to just go through the membrane, and there's an ATP synthase that will help to make ATP with the hydrogen flow. This whole thing is the chemiosmosis. The hydrogen ion is going to get attached to NADP plus to form NADPH. The photosystem 2 is the strongest oxidizing agent. It's a strong oxidizing agent. The PS2, the chlorophyll inside chlorophyll A, because it takes away the electron from the oxygen, although we know that oxygen is really a strong oxidizing agent. Electrotransport chain makes the ATP, and the PS1, the photosystem 1, is responsible for making the NADPH. The light-independent reaction is happening in the stroma. This is the time when the carbon dioxide is going to be reduced and carbohydrate is going to form. The carbon is going to fix, reduction happens, and regeneration, the RUBP. RUBP is a chemical that is necessary for the Calvin cycle. The carbon dioxide is coming into the carbon fixing place, and the RUBP combines with the carbon dioxide with the help of Rubisco, an enzyme. The carbon dioxide in this case is the limiting factor. Reduction happens. This is when electrons are added and protons are added to the carbon. Requires energy and requires the NADPH. They are both coming from the light dependent reaction, and the end result is 3-phosphoglycerate and from this 3-phosphoglycerate goes the glucose. In arid climate, the different topic, arid climate, the dilemma is huge for the plant. Make food or conserve water, because it's all about the stoma or the stomata. This is the dilemma. If they want to make food, they have to open their stomata to get the CO2 in. If they want to conserve the water, they have to close the stomata, but with that, no carbon dioxide will get into the leaf, so the oxygen concentration will increase due to the concentration will increase. If the oxygen concentration increases, photorespiration will take place. Why is that? Because the oxygen will combine with rubisco and produces a 2-carbon compound and uses up ATP and oxygen. Produces CO2 but no sugar at all. So for cellular respiration, there's no sugar. Now the stoma opens usually at dawn and closes at nighttime. It shows a 24-hour circadian rhythm. The stoma closes at high temperature. Additionally to nighttime, the stoma can close at daytime when it's high temperature, or during drought, when the plant battles with water content. Evolutionary wise, let's check the photorespiration. It can happen because rubisco is non-selective enzyme that much, so when it used to be high carbon dioxide concentration in the atmosphere and low oxygen, it worked really well with the carbon dioxide. And also the light reaction products need to be broken down because they might be devastating for the plant. We have 3 different types of plants based on how they got accumulated to the environment. The normal plants are the C3 carbon plants. They have the Calvin cycle, totally normal. Carbon dioxide goes in. They will have a 3-phosphoglycerate molecule and the sugar comes out. Photorespiration happens at high temperature and dry climate. That's really bad for the plant. We said that they cannot make any sugar molecules. So the evolutionary wise, we have the C4 plants, for example, sugarcane and corn, that they really can withstand high temperature and dry climate. They have here is the leaf cross section, the upper epidermis, lower epidermis, and the vascular bundles, the xylem and the phloem over there. And around the xylem and phloem, we have the bundle sheath cells. These cells are going to be the cells that they will perform the photosynthesis. In the mesophyll cells, the carbon dioxide is going to be fixed, so carbon dioxide goes into the mesophyll cells and will combine and form a C4 material inside of the mesophyll cell. They will combine with this carbon 3 to form carbon 4, and the carbon 4 is going to be transferred into the bundle sheath cells, and that C4 over there is going to be split again to C3 and carbon dioxide, where the carbon dioxide can freely enter the Calvin cycle. So the point is, with low carbon dioxide concentration, it still can supply carbon dioxide to the cells in the bundle sheath to do the Calvin cycle. So the problem is that to make these C4 and C3 compounds that can transfer the carbon dioxide, it requires ATP, so additional energy needs to be invested into the process. But it's really good because there's no photorespiration for that plants, or very limited, if at all. The other type of plants that just got used to the high temperature and dry climate without photorespiration are the chem plants, the pineapple and the cacti, for example. What they do, it's nighttime, the stoma opens, and the carbon dioxide will combine with a 3-carbon material, and they form an organic acid that is 4-carbon compound. On the other hand, at daytime, when they can close their stoma, they get the carbon dioxide off of the carbon 4 compound, the organic acid, and the carbon dioxide can go into the Calvin cycle. So what they do is this process separates the fixing and the using in time, so nighttime and daytime. On the other hand, the C4 plants, the fixing and the Calvin cycle, these processes are separated in place. One happens in the mesophyll, the Calvin cycle happens in the bundle sheath cells. So the good side for, so yeah. So for the chems, the processes are separated in time. The good for C4 and chem cells, that the photorespiration really decreases. So photorespiration producing carbon dioxide and no sugar will decrease. And the bad part is ATP is needed for fixing the additional compound. That's all.